Thank you so much, Dr. Kumar. Great, thanks. Just before I get started, just wanted to thank the organizers for having me at another amazing APO session here in India. Great to be here. And I have no disclosures to make. So quite a lot of what we know about elevated IOP and glaucoma comes from multi-omic studies or like single omic studies. So we know there's like hundreds of genes that are involved in both elevated IOP and POAG, and they are expressed in a highly tissue-specific manner. We know that age-related driven epigenetic status changes around those genes can at least in part influence and dictate the late onset that we see for a lot of glaucoma cases in the population. And we know that metabolism, specifically photooxidative stress and lipid metabolism and others are important to the homeostasis in the anterior segment and trabecular meshwork. Now, talking about multi-omics, like often the base layer of the omic stacking are the GWAS or the genome-wide association studies. There are several of them that have been published over the years. The most recent one is something we are still working on, and we put together a cohort of 350,000 individuals, more or less, with both IOP measurements and SNP data. And as usual, the more power you have, the more you know about the genetics of a disorder. So we've identified a couple of dozen actually new genetic regions that are annotated through those like vertically oriented labels in that figure. We also see like higher levels of association, but these are known loci, some of which are actually still shown on this figure. Now, that sort of genetic knowledge is useful to make predictions. And although imperfect, our genetic predictions are getting better, and we have this unprecedented ability, and still it's work in progress, but to identify individuals who are going on to become glaucoma patients or who'll stay controls for the rest of their lives. What does that tell us about the gene functionality? Something but a little. So we know, for example, there are a few recurring themes among the GWAS association studies. We know, for example, just to cite like a few random annotation labels, that angiogenesis, cell adhesion, extracellular matrix, lipid metabolism, and much more are overrepresented among the top GWAS hits. But we know very little about how those genes link together in a coherent cascade to actually elevate IOP and cause myopia, or conversely, to contribute to good health in the eye. And this is not just genetics data. Like every single large omics biological data analysis has the same limitation. So these findings tend to be associative in nature, so they tell us little about causality. They are annotation or enrichment-based, and they just heavily rely on co-occurrence and co-localization with little thought about causality. And these are all good questions that integrated omics approaches can answer. And specifically here, I'm just going to try to focus on the obvious ones, like how are the genes and proteins working together to maintain health or to increase glaucoma risk? And are there any specific biological cascades that contribute to those conditions? And again, the implicit question is if they can be exploited for new therapies or otherwise in the clinic. Just a sort of like so essentially just to bring you back to like biology 101 on transcription. We just heard from the excellent talk that Dr. Chakrabarty gave that there are so many genes harboring mutations that can cause very dramatic, life-changing, early onset and clinically severe glaucoma in early life. And those genes tend to be in the protein coding parts of the gene, which are shown here, like using the red symbols at the bottom. But the SNPs that we identified through genome-wide association studies tend to cluster at the other part of the gene earlier on in the sequence, around the enhancer and the promoter region. So about half of the SNPs that we've identified for our GWAS of intraocular pressure and glaucoma are actually in that region, and they have significant effects over the level of the mRNA that is being transcribed, adjacent to those like sites of association. Now, the same gene, we see an overlap, so the same genes can harbor mutations, the same genes can actually have EQTL effects associated with glaucoma and IOP, and here I'm just bringing a few examples. So TAC genes has important SNPs that are common in the population that are associated with changes in the transcription process, and so is LMX1B, the gene behind nail patella syndrome, and the ABO SNPs apparently are associated with both IOP and have EQTL effects. So this is all stuff we knew. Like, how about, so the real question here is like, is there any remote ability from those genes? So can they actually change something remotely elsewhere in the genome, something that they don't do physically, but they do through interactions, like protein-protein interactions, somewhere, if they can actually leverage any biological cascade to change something down the stream. To do this, to answer this question, we actually assembled a large data set. Here, just reading like mistakenly, no, it's not mistaken. So here we just like assembled this large data set, like thousands of individuals with both peripheral plasma protein measurements and SNP data. And we found that not just in the context of IOP and glaucoma, but some of the major hubs of interaction, trans interaction, like distant interactions, were just revolving around the ABO gene that we just mentioned is important for the development of like elevated IOP and glaucoma. So the ABO, like the blood group locus, had some really outrageous like levels of association and all over the genome. More in the context of a glaucoma / like elevated IOP, the locus at the ataxin-2 and SH2B3 genes that are very like next to each other on chromosome 12 is also very important. Of all the genes that participate in mechanism we believe that are associated with IOP and glaucoma, those two genes are responsible for 99% give and take of the interactions that we see. And this is a simplified version of the same chart. Essentially, every arrow is a significant association between a SNP and a protein. And the genomic loci where those SNPs are located are actually shown like in faint blue. And the genes that encode the proteins those SNPs are associated with are actually highlighted in yellow. Unfortunately, those arrows have slid a little. But again, a vast majority of those interactions still revolve around ABO and the locus on chromosome 12. There are a few limitations to what I just mentioned. So this is like an oversimplistic representation of the reality. So those arrows often involve like tens, if not like hundreds of other proteins along the way. And blood proteomics can be a blunt tool occasionally. There's good reason to use it, but there are limitations. Typically, when you do plasma proteomics, you only focus on very abundant proteins that you can see, but that kind of excludes like transcription factors, which are important in their regulation. And in the blood, you actually measure pretty much what's washed in the plasma, but you don't have the tissue specificity you may need. Therefore, transcriptomics can actually be a more sort of delicate and elegant tool that we decided to use as a next step. And I'm really grateful to Dr. Chingyi for explaining the principles of the Mendelian randomization in a much better way than I would have time to. But basically, the method involves like instruments, like SNPs are used as instruments. They have a certain effect over an intermediate parameter of some sort. And then we are interested in understanding better the relationship between that intermediate parameter and outcome of interest. Now, again, like those intermediate parameters and outcomes of interest can be anything we want, but in this context, I'm going to talk about both the intermediate parameter and the outcome being levels of transcribed mRNAs. And I knew that it would be like time would be like very short, but to do like using those so simplified Mendelian randomization to mass scale and using some pretty stringent false discovery rates, we just discovered quite a few relationships that we believe are actually quite convincing. And here I'm just like showing like a part of the picture that is like slowly emerging, and we actually see even at a transcription whole blood transcriptomics level that the ABO locus has the ability to regulate the TEC peripheral expression in the blood. In some tissues, also like myocillin, somehow in the blood, but a lot stronger in other tissues that were available to us. They don't seem to be immediately related to glaucoma, but we saw very strong regulatory effects in skin. And those interactions, again, like those arrows, in a way, like hide the true complexity of those interactions. So they are an oversimplified way to represent what we believe to be the reality. So the takeaway sort of message is that the ABO have very significant trans-EQTL and PQTL effects that were some of the strongest that were ever detected on humans. The ABO locus may be a regulator of pathways that are important in angiogenesis, both systemically and in glaucoma genesis narrow context. The ABO blood group B, or like SNPs that are in strong LD with the ABO blood group B, tend to elevate the TEC expression in peripheral blood and other tissues. and they have like remote regulatory effects over other glaucoma genes. Attacks into alleles, the attacks into alleles that tend to be associated with increased levels of IOP tend to decrease attacks into expression, and they tend to increase the myocilin gene expression. And this is again like a partial view of what is slowly emerging. So many funders, like collaborators, institutions I wanted to thank, and so many of you are actually in this room, so thanks for your support. But this photograph, again, is a lot of people who are just doing the IgGC genetic analysis, and I'm grateful to all of them. Thank you so much again for your time.
Thank you, Professor HYSI. So questions, comments, please. Yes, Shubham.
Circulating plasma, because you're looking at a very focused disease. So,
yeah.
Because again, as you would know, the abandoned proteins would be much higher there, like albumins and other things would go up. But so the second related question was the ones that are surrounding the ABO locus, were they the low abundant proteins that you got?
The protein, plasma proteins, yeah. They were like low abundance. They were high abundance. In the plasma, we only measure those ones that are above a certain threshold.
Right. So the first question was the rationale of using plasma,
right.
Circulating plasma.
So two very basic sort of reasons. Like first of all, that is like availability. It's cheaper, it's easier, it's more practical to actually do it on plasma. And there's like a hidden sort of like a purpose, if you will. Identifying biomarkers. Say that you identify this biomarker that diagnoses glaucoma, like at birth or anything. It's not going to be like practical to actually get like an eye biopsy at birth or anything. So you actually kind of ideally want to do that in plasma. So that's kind of like a hidden wish we had.
Thank you. So, Jenny.
Pirro, thank you for that really super interesting talk.
Thanks.
I have a question more around methodology. And that is, in your initial analysis where you looked at all of these, protein interactions and kind of funneling in on these two hubs of ataxin-2 and ABO, how do you account for, or how do you correct for, or how do you remove the bias of proteins that have a lot of protein interactions? So for example, ataxin-2 does a lot of stuff in the cell. And, so just sort of naturally, if you don't correct for that in some way, those are going to be identified as important hubs because they're pleiotropic. They just do so many different things. Where there could be a very specific interaction, just like one interaction between two proteins that's critical to glaucoma, but wouldn't have survived your analysis because it wouldn't have formed those two hubs.
Again, it's going to be like the first part of the quick answer is going to be like how we define those. What is an interaction? Again, like to me, this is actually stuff that we are exactly looking for. Essentially, what ataxin or ABO can actually do downstream, like what those SNPs will never interact directly with the transcripts, like somewhere like in a different chromosome. But it's essentially through the ataxin protein doing something else elsewhere in the body that we're getting those associations. So essentially, that was kind of like the purpose why we did that as opposed to like a byproduct. But again, like think about bias. Bias could be like population stratification. Again, like blood group is we had to, part of the reason why we're doing Mendelian randomization, we're doing that at a much more sort of like careful way than I showed here, is that again, like that could be like a huge source of bias. But I'm not very worried about the multiple sort of pleiotropic effect ataxin has, because essentially there has to be a cascade underneath that, and that cascade is essentially something that we want to characterize.
So I have a question, Professor HYSI. So if I understand it correctly, what you're suggesting is that the blood group is a platform, and then if you have B blood group with TEC, then you get glaucoma, right?
You're more predisposed in average to have glaucoma.
Okay. So my question is, are there any other eye disease or human disease which has such kind of a link?
Lots. Again, lots. Again, like gastrointestinal cancers are related to blood ABO blood groups. Like
stomach.
Lots of them.
Yeah,
we
that's
see,
a very long known
everywhere.
fact,
Like
yes.
a ton of immune responses.
Okay.
Now, again, I specifically focused. It's a much longer story, but very briefly here, I just focused on that because that's a massive effect. What we are seeing actually, those SNPs that we use as instruments and those effects that we see, they actually change from tissue to tissue. So we actually see, like ataxin interaction, we see the same ataxin effects over somewhere else, but actually the genes, the SNPs that we use as instruments are slightly different. So they're actually located somewhere else in that promoter. So that shows like a certain tissue-specific activity that we found very interesting, but I didn't have time to show here in full.
Okay. Very interesting. Thank you, Professor
Thank you.
HYSI.
Thank you, Pirro.